Hi everyone, and thanks so much for tuning in to Science at Home. I'm Kris Thunhorst, a staff scientist with 3M Company. I also work in our Visiting Wizards program, where science is fun, and in our Young Scientists lab. I enjoy bringing scientific principles to kids and making them exciting and real. It's good for adults too. Today, what we'll be talking about are acids and bases. When most people think about acids, they think about things like lemons or limes. But did you know that even water can be acidic or have different levels of acidity or basicity? Have you ever heard of acid rain, for example? That's sort of an extreme example, but water can vary in acidity. In fact, I have flowers outside my house called hydrangeas, and the flower color depends on the level of acidity of the soil. So I can add chemicals to the soil to make those have blue flowers or to make them have pink flowers. So that's a real life example of acids and bases. Today we're going to do some experiments at home. Acidity and basicity is measured on a scale called the pH scale. It ranges from 0 to 14. 0 is the strongest acid, and so acids go from 0 to 7. Bases go from 8 to 14, and 14 is the strongest base. Our bodies, we also have a pH associated with our bodies, and it likes to be close to neutral. Scientists have discovered ways to visualize this scale of the pH range, and it's through chemicals called indicators. Indicators change color depending on the level of the acidity or the basicity of the items that they're measuring. So today, that's the approach that we're going to use with materials that you have at home. One of the things to keep in mind, though, before we get started on our experiment is safety. As scientists, we're always concerned about safety. A lot of times you'll see us wearing safety glasses and gloves. Today, because of most of the materials that we're dealing with are not hazardous, we won't wear any gloves, but we will always wear our safety glasses. And the other thing to keep in mind is, even though we're doing these experiments at home, adult supervision is required. So what we'd like to cover now are some of the materials that you'll need to do this experiment at home today. First, we have a blender, and in that blender, you'll notice I've already put some pieces of red cabbage, so we'll get to that in just a second. So you'll need a head of red cabbage, your blender, and then some water. And what I've understood from the experiments is that distilled water actually works the best. You'll need a variety of containers. These don't have to be clear glass containers like I've got. I'm doing that because it's easiest for me to share with you on video what these things look like. You could use anything from as simple as an ice cube tray to glasses from your cabinet if you like. If you're going to make your own juice, like from your citrus, your lemon or your orange or lime, you might like to have a juicer on hand, but a fork works just as well to get that juice out of your lemon. You'll also need to have a container, a larger container than the ones that we've got to make our colored solutions in. And this one will be to make your indicator solution with. And with that, you'll also need some sort of a strainer. So I am using this little screen basket with a coffee filter that you'll see today, how we'll use that. But if you don't have one of these fancy little screen things, that's fine too. Just a regular funnel or something else will work. So you can just put your coffee filter in that when we make our indicator solution, which we'll do in a little while. Then I like to have a few spoons on hand because I like to stir the solutions. And then there are all the materials that you want to test. So I've got some labels on these, but we've got lemon juice and orange juice that I juiced up last night, so we'd be ready. I've got some sparkling water here in case we want to test the pH of that. I've got some just white regular vinegar. We've got some table salt, just regular Morton table salt. We've also got some baking soda, and then the special ingredient that I found in the cabinet last night that I thought might be fun is we've got some Pepto-Bismol, so we can see what happens with that. So you might think about what materials you'd like to test the pH of and have those all on hand for your experiment. Now, I'd like to make the indicator solution, so it's going to get a little bit noisy. What we do to make the indicator solution is we take 2 leaves off of this red cabbage head, and I've done that ahead of time. I've got one already in there, and I've got a couple more pieces here waiting to go in. And those are going to get stuffed down in your blender. And then for each leaf of red cabbage that you use, you're going to want to measure about 1 cup of water. So this measuring cup that I've got is a 1 cup measuring cup. So we're going to fill this twice from our distilled water. Okay. And then we're going to close up the blender so that we don't get things sprayed all over. And it's safer that way too, so that we don't get ourselves into the sharp area of the blender. And then we're going to want to blend this for 30 seconds, and it'll be a little noisy, so you won't hear me for a couple of seconds here while the blender is making a bunch of noise. So I was counting in my head, that seemed like it was about 30 seconds. So I'm going to go ahead and open up the blender. And this is a little bit messy. There's some pieces, we call this cabbage soup. I don't think it probably tastes very good, but it's going to be really awesome for our science experiment today. So here I'm going to put together my little screen in my larger glass dish, and I'm going to set my coffee filter in there. And then I'm going to take this solution that we just made and pour it slowly into the filter, because there's a lot of solid in there now, a lot of little pieces of cabbage leaf. And so the water will go kind of slowly through that filter. So what we have here is our indicator solution. And remember, those indicators are materials that change color to indicate whether things are basic or acidic. And so now what we want to use is this very special homemade indicator solution to ask questions about those common household items that we have here. So what I'm going to do first is to pour some of that indicator solution into these glass containers. And I'll just start by pouring it into 4 of them. And you don't need a whole lot, as you can see, which is why an ice cube tray would also work. There we go. And we'll save the rest, because I think we'll have more questions. And so now you've got all these different materials, and like a good scientist, you might say, what do I expect to happen? What things do I expect to be acids, and what things do I expect to be bases? And you can separate your materials based on what you think is going to happen. So if we were to do that, I don't want to really spoil the surprise. I might let you guys do that on your own. But we already talked about the citrus being acidic, so we might expect the orange juice and lemon juice to be acidic. So those might go in one group. If you guys, and maybe vinegar as well. So those 3 things might be acidic. We have some questions about salt. We're not maybe quite sure what would happen with salt. The same thing might be true with sugar. We might not know what would happen with sugar. What about hand soap? We don't know what would happen with those. So those might go into an uncertain or maybe a neutral pile. And then we've got other things. We've got some carbonated water. We might not know what's going to happen with carbonated water. And we also might not know what's going to happen with baking soda, although we might suspect that this one would be a base. And then the last one that's really fun is Pepto-Bismol. So last night I took a spice grinder, so some of you might have these, and I ground up a Pepto-Bismol. This is where you need your parents' help. But I ground up a Pepto-Bismol pill, and interestingly, they're as pink on the inside as they are on the bottle. But we might have questions about what Pepto-Bismol does. But if we think about the purpose of Pepto-Bismol, it's to reduce stomach acid. So in that case, we might think that it has some basic properties. So that can maybe go in the same category as our baking soda, as we suspect that that might be a base. So now we've got all our materials set up, we've got our hypothesis. Now we need to do a test and actually figure out whether our hypothesis was right or not. So let's go ahead and do that. Let's start with vinegar and see how that works. What happens with vinegar? So I'm going to take one of the bottles with our indicator solution, and I'm going to pour just a little bit of vinegar in there. Wow, you can already see that changing color. At least I hope you can see that on the video. If I hold it up next to one of the other ones, maybe it's easier to see. And that's one thing I do recommend. If you would leave one beaker that has just the indicator solution in it, sometimes it's easier to compare how that color change is happening. It's really important to keep track of what you're doing. So here, I've got some extreme notes. They're good if they get wet. So I'm going to write on there vinegar. And I'm just going to stick that on the container or in front of the container where we added the vinegar. And that way we can remember what we added. So that was something that is acidic because this particular indicator, the cabbage leaf juice, when it encounters something that's acidic, it turns red. Or on that red spectrum. You'll see that change gradually from that purple to red. So let's try something that we suspected might be more basic and see if that's any different. So this is the baking soda. So I'm going to take a clean spoon and I'm going to take a fresh beaker of our juice and I'm going to take just a little bit of baking soda. And this is, hopefully you guys can see this. I'm going to put my hand underneath, but it's just a little bit of baking soda on the end of that spoon. And I'm going to set that into this beaker and stir it. So it's a solid. So you can see the color change isn't happening right away. But like all good scientists, we're going to give it a minute and have some faith. And as we stir, we're noticing some differences happening here. So in just a minute, I'll pick up the control, the one that we didn't put anything in, and hold it up next to it so you can see. But what you notice is that it's changing color, isn't it? So it's moving more toward that dark blue color. And so this particular indicator, this cabbage juice, when it encounters bases, it turns blue. So let's set that one next to our vinegar, and you can definitely see that there's a big change there, even though they both started from that same solution. So let's go ahead and label that one too, because we don't want to lose track of what we're doing in our experiment. So that's baking soda. Okay. And then what should we do? I mean, there are so many choices. Let's try sparkling water. We might not know what's going to happen with sparkling water. So we're going to go ahead and open that up and see what happens. So this is our third jar from our original solution. I haven't added anything to this one until now. And now I've added sparkling water. Let's see, I'm going to add a good amount of sparkling water to this one and stir that one up. So when we compare that to the vinegar jar, what do you notice? It's not quite as red, is it? But it's not quite as purple as how it started either. So what does that mean? Maybe it means that the carbonated water is a little bit acidic, but not as acidic as the vinegar that we added. And did you notice? We added a whole lot more of the carbonated water. So this colorimetric is what we call this technique when things are changing color as they're exposed to different pHs. This colorimetric technique can tell you which things are stronger and weaker acids or bases. So let's close that water up so I don't knock it over in the rest of the experiment. It's nice to have a tidy experiment area. And so that we don't forget, we're going to write down that that's sparkling water too. And then, let's see. Let's try, this is the orange juice, and this is the lemon juice. So let's try the orange juice. And this is juice that I squeezed from an orange last night using this juicer, but it doesn't matter. You could use orange juice from your fridge as well, whatever you have. So I'm going to pour a little bit of this orange juice into this beaker of original solution. And you can already see it changing color. In fact, before I mix it, I can kind of see it changing color from the bottom up. So that's a really cool thing. I have almost like 2 layers here before I mix it. It's a little bit darker purple on top and a little bit more reddish, pinkish on the bottom. So I'm going to stir that up a little bit until it's all uniform. And we've got something that definitely changed color, right? I'm going to take a little bit more of our original solution and pour it into one of these other glasses so you can see a comparison. So in my right hand here is our original solution, and here's the one that we mixed with the orange juice. So you can definitely see that it changed color. And then we might ask ourselves, is that a stronger acid or a weaker acid than the vinegar? And when we look at it, it sure doesn't look as red as the vinegar does it? We also have to be, if we wanted to do a really good scientific quantitative experiment, we would measure how much of these things that we're putting in, and we would add equal amounts, and that would actually give a more scientific result about the strength of that acid or base. But if we compare it to the sparkling water, it's more similar maybe to the sparkling water than it is to the vinegar. So let's go ahead and write down what that one was as well. I'm going to pour more of our indicator solution into a couple of these glasses. So these are both original solution that hasn't been mixed with anything yet. And then let's try the Pepto-Bismol and see what happens with that. So I'm using a clean spoon again. That way we don't get contamination of one chemical into another. So we're actually understanding what the true result is of that particular thing that we're testing. So here's my crushed Pepto-Bismol. And I've got, again, just a little bit of that on a spoon, kind of like the same amount as the baking soda. I'm going to go ahead and put that in. And remember, it's a solid, so right now that solid's floating on top of the water, so we can't get a good reading. So I'm going to go ahead and stir that in, and it kind of still wants to float. So I'm going to mix it up, give it a little time here. If you track this over time, you might see that it changes over time as well. But let's go ahead and hold that one up. I hope you can see on the video, but I can certainly see that it's definitely more blue than the solution we started with. But if we compare that one to the baking soda, my goodness, look at how dark the baking soda has gotten already. So it's not as strong of a base as the baking soda is. I'm going to go ahead and write down that that's Pepto-Bismol. Okay, we'll label that one too. All right, and then let's try the table salt and see what happens. And I wonder if any of you at home have a theory about what's going to happen when we add this table salt. Let's see. Is it going to turn blue? Is it going to turn red? Is it going to stay the same? So let's give it a try. I'm just going to take some of this table salt and pour it in. Oops, I'll use a clean spoon again, so no contamination. And again, since this is a solid, we want to make sure that it dissolves in order to get the real answer of the test. And what I'm noticing is, I know these aren't quite the same kind of glass, but there's not a lot of change in the color of the solution when you put the salt into the indicator solution. And what that means is that salt is not changing the acidity or the basicity of the solution. It's merely dissolving in that water. So that's salt. I'm just going to write that one down as well and label it. And so now, if you look at what we've done, you can see a whole variety of different colors. We've not quite made a rainbow. Maybe that can be one of your challenges at home. But we've got a nice variety of different colors, and we've had a lot of fun doing science experiments at home. I hope that you have learned something today about acids and bases. I hope you really enjoyed doing these experiments. And I would encourage you to check out all the other great videos from 3M Science at Home. I've got some wonderful colleagues who've really put together some thoughtful content for you. So thank you again so much for joining, and I hope you have fun. Have a great day.